Today I'm going to talk to you about antibiotic resistance in bacteria. And the way I've framed this talk is we'll talk about more about the problem of antibiotic resistance. And then we'll talk about some of the mechanisms of resistance. And then briefly I'll tell you about what we do in our lab. And then finally, a big question, what is actually contributing to antibiotic resistance? To begin with, let me just take you back about 100 years. So in 1900, what was actually happening was, in terms of infectious disease, someone like me could be shaving, cut themselves while shaving, get infected and die because there was just no cure for bacterial infections. So you could be working in your backyard, get cut, get infected, and basically end up dying. So New England Journal of Medicine is celebrating 200 years this year, and they actually put up some really nice data showing how we as humans were dying in 1900 and how we are dying today. So the causes of mortality. And this data is from US, but what I would like to highlight here is that most of the deaths in 1900s, you see that these were from infectious diseases. So right here, here. What has changed since 1900 is that, at least in developed world, is that mortality due to infectious diseases has gone down dramatically. It has changed, it has really changed the way we die. And and it has really increased. One of the reasons why we are living longer now is because we have sort of, well, at least we thought that we conquered infectious diseases. And antibiotics are one of the key factors contributing to this. Vaccines are another factor. We are getting vaccinated against a lot of these infections, but antibiotics definitely have played a big role. And this all started with this gentleman here. This is Alexander Fleming, who in a July afternoon in 1928 went to his lab in England and then found a mold growing on his bacterial plate, and he actually noticed, he was a very observant scientist, he noticed that the mold was actually inhibiting the growth of bacterial cells that were plated on his plate. And that was the start of modern era of medicine when it comes to antibiotics. The first patient, civilian patient to be treated by antibiotics was in a hospital in New Haven. This was in 1948. A 19-year-old woman was in critical condition. She has had a miscarriage, and she had streptococcus infection, a bloodstream infection. Her fever was spiking up to 107 degrees Fahrenheit at times, and there was just no way to cure her. They had tried sulfa drugs, which were already in market at that point, and basically physicians were waiting for her to die. Turned out one of the physicians attending to her knew this gentleman, Howard Florey, who was visiting US from England, and he had brought 5 grams of white powder in a vial, trying to convince pharmaceutical companies to mass scale the production of that product because this was penicillin. So this physician called Howard Florey and he said, well, can you give us some of your powder? And he said, okay, I'll give you 2.5 grams of that. So he gave him half of that, and they really didn't know what to do with that powder. So they dissolved it in water, and they injected a bit of that solution in this patient, and basically just watched her for adverse reactions. And there were no adverse reactions. They just injected the whole thing. And then the next morning, they actually noticed that fever had subsided. And this was the first patient in a civilian hospital treated by, her name was Ann Miller. She happened to live for 90 years. She actually died 3 years ago in New York. So that was the first instance, and that actually changed everything. From there on, science like this were becoming more and more common. People just thought that you could treat anything with antibiotics. And what followed was what we call as golden era of antibiotic discovery. So in, about 20 years after penicillin was first discovered, we started finding a lot of different compounds which had antibiotic property or antimicrobial property. The perception was that we really don't have to worry about infectious diseases anymore. And in fact, in 1967, William H. Stewart, who was a Surgeon General in Johnson administration, made this famous comment, or infamous now, that it's time to close book on infectious disease. We really don't need to worry about infectious diseases. We don't need to fund research on infectious diseases because it's done. These are things of past. So just around the time he made this comment, things started changing. 1958 was the first time a beta-lactamase enzyme was found in a hospital in Japan. So this was the enzyme which could inactivate beta-lactam antibiotics. It was found in an organism called Serratia marcescens. And then as you notice, what happened after that is that our search for new antibiotic compounds kind of just started to dry out. We were finding less and less new antibiotics that were effective because the resistance was increasing and new antibiotics were not really easily accessible or, well, not easily discovered. So things sort of started going backwards right around the time Dr. Stewart made that comment. And this data here sort of just shows what's the global picture of antibiotic resistance. So this here is data from European CDC. We're just looking at a map of Europe. And if you look at this panel right here, this is fluoroquinolone resistance in E. coli. And within 10 years, the resistance is from up to 10% has gone up to more than 50% in E. coli. So 50% of isolates of E. coli in some of these European countries are now resistant to fluoroquinolones. So resistance is growing, and this is just in 10 years. Close to home, it's not much different. We were actually just talking about gonorrhea over lunch. A bunch of microbiologists, what else? Nothing interesting than that. So this is data from Ontario. And what we see here is these bars are showing number of isolates of gonorrhea that were reported or identified. And you see that number of isolates have more or less stayed the same over years. What is alarming is the percentage or the fraction of isolates that are now resistant to quinolones. So now we have, as reported in 2006, so this was 5-6 years ago, up to 1/3 of the isolates from Ontario gonorrhea isolates are resistant to quinolones. Actually, CDC came out and identified geographical areas in North America where they are just saying do not use fluoroquinolones for gonorrhea treatment because resistance is really, really rampant in gonorrhea. So what has happened as a result of increased resistance to antibiotics is that World Health Organization has now recognized antibiotic resistance as one of the greatest threats to human health. And it's calling for more research, it's calling for better infection control practices, it's calling for prudent drug use, it's calling for better quality of drugs, it's calling for strong surveillance, and it's also calling for stronger commitment from government agencies, from researchers, from physicians, from clinicians, in order to deal with this problem. Because the fear is that it's not just the infectious disease. Just think about what other kind of medical practices would be effective if antibiotics were ineffective. So right now we use antibiotics post-surgery in transplantation reaction or post-transplantation reaction. And all of these processes will be adversely affected if there were antibiotics or there were no antibiotics to actually cure bacterial infections. So the question is, well, okay, we know that resistance is really rampant. We know that old antibiotics don't work as well. So why not just go out and find new antibiotics? And as we were, again, just discussing this over lunch, one of the problems with finding new antibiotics for treating bacterial infection is that there are very limited number of targets within bacterial cells that can be exploited by antibiotics. Now, when you're looking for an antibiotic, as the name suggests, it's against life. So antibiotic is a poison. So we need to find a poison, we need to find a molecule which is selective against bacterial cells, but does not affect us. And when it comes to those sort of targets, which are specifically present in bacterial cells, but not in humans, there are very few of those. And it seems like we have pretty much exploited every single target that's out there. We still see few reports coming out where they identify new targets, but those reports are very, very rare. So that's one problem. We don't really have good targets for antimicrobials. The other problem is, which is just completely commercial nature, is that it's very difficult to convince big pharma now to go and find new antibiotics. About 10 years ago, there were 14 big pharma companies that were involved actively in antibiotic discovery programs. And today, there are only 4 of those. And one of the reasons for this is, as this chart is, I borrowed from Pfizer, and sort of shows how painful the process of finding any drug and putting it into the market is. Takes about 15 years from lead compound to put the drug into the market. When it comes to cost of antibiotics, putting those in the market, it's about $1.2 billion. And with current patent laws, you have about 7 years to recover this $1.2 billion. So unless you get about $1.4 billion back from marketing of this drug, it's really economically not feasible if you are in business of making money. Now the problem here with antibiotics is that if a pharmaceutical company is committed towards finding a new antibiotic, spends all this money, has all the expertise, puts it on the market, and the drug is really, really effective, then our advice to clinicians is, well, don't use it. Because if it's an effective drug, you want to put it on the shelf and use it only when everything else fails. That means there is no money coming from that drug. So pharmaceutical companies are primarily interested in treating chronic conditions. So a drug for cholesterol is something that a patient would use for the rest of their life, and it's a good source of revenue. So that's one of the problems with in convincing pharmaceutical companies in discovering new antibiotics and putting them out in the market. So I'll change gears a little bit now. So I'll tell you a little bit about what we do in my lab. And we are sort of, at our small level, we are sort of trying to contribute towards finding novel ways of treating bacterial infections. So this is my laboratory group from past summer, and there's always a large turnover. So some of these students are already gone. Leah is a fourth-year med lab student who's currently doing her clinical practicum. But these 4 are my graduate students who are basically in the lab all the time, or they should be, or I hope they are. So one of the organisms that we work with is Acinetobacter baumannii. Acinetobacter is a gram-negative rod, as you can see here. It routinely shows multi-drug resistant phenotype, which means that it's resistant to at least 3 classes of drugs. And whenever we find a multi-drug resistant Acinetobacter baumannii, the only treatment option left is colistin, which is an old drug. It was used in 50s, and then the usage stopped because of high nephrotoxicity of this drug. So it causes a lot of damage to kidneys. And that's probably the reason why we don't have colistin resistance right now, because we haven't been using this drug for past 40 years. And tigecycline, which is a newer drug, it's a glycocycline drug. It's a very new drug, so resistance rates are low. So these are the drugs we use if the organism is multi-drug resistant. The resistance shown by this organism is very rampant, very high, and that's why sometimes it's referred to as gram-negative MRSA, just because of its widespread resistance. The organism gained a lot of interest in past 10 years or so, because what was happening was a lot of our soldiers who were coming back from war zones, from Iraq and Afghanistan, were coming back with infections with Acinetobacter baumannii. So these are 3 Canadian soldiers who came back in 2004, I believe, and these are the wounds that they would typically come back with. So a shrapnel wound, amputation, a bullet wound. And what would happen is that these wounds would get infected by Acinetobacter baumannii. So what was happening was that they wouldn't really die from the wound at the battlefield, they'll actually die from infections, which they would pick up in the hospitals. So this is where interest into Acinetobacter really became rampant. And what has happened since then is that the organism has spread quite a bit in our hospitals in North America. So now in US, in Canada, we see Acinetobacter a lot. And you can see here, this data is from US, and this is data for carbapenem resistance in Acinetobacter, one of the last line drugs that's used for Acinetobacter infections. Here you see that rate of resistance, this lighter shade of orange indicates less than 10% of isolates were resistant to carbapenem, and here we have almost 50% of isolates which were resistant to carbapenem. Again, happened in 10-year period, just around the time when these wars started. And what is happening with Acinetobacter is that there are more and more cases now where a patient comes to the hospital, picks up infection, and these are generally hospital-acquired infections, so picks up infection and basically dies because there's just no drug to treat the patient. This is a patient from Germany. This was a cancer patient who had pneumonia, so he basically walked into the hospital complaining of pneumonia, or he was diagnosed with pneumonia, and he was hospitalized because he was a cancer patient, and then they started imipenem on the patient. These numbers are the number of days that he was hospitalized for. This is a 76-year-old patient, and they started with imipenem, the infection didn't go away, then they started meropenem, tobramycin, ciprofloxacin, sulfamethoxazole, and the infection never went away. Colistin, then went back to meropenem, and after 96 days, they basically figured that there's nothing they can do, and end-of-life decision was made. So this patient ended up dying in the hospital. What this is actually showing is what we used to see in year 1900. You get an infection, and you just have no way to cure this. One thing that we did, or what they did here was, and this is just going back to what sort of technological options we have these days, was these isolates. These letters here indicate the days when they isolated the organism. And what they did was they took these isolates from different days, same organism, and they sequenced the entire genome just to see what is happening at the genome level in this organism. And what they found was that one of the key factors was overexpression of certain proteins called efflux pumps. So efflux pumps, and I'll explain in a bit what these are, were overexpressed at this point. Isolate D was the first isolate which expressed efflux pumps. The organism became multi-drug resistant, and that resulted in accumulation of other resistance mechanisms. So organism is really smart. It's actually evolving inside the patient as we are administering antibiotics to the patient. So it's keeping ahead of us here, just evolving mechanisms to to derive antibiotic resistance. So what are these efflux pumps? So let's just for a second believe that this is a bacterial cell, and this is the antibiotic. So antibiotic molecule needs to come inside the bacterial cell and act on the target. But these efflux pumps are the proteins which are associated with bacterial membranes. So here we have proteins attached to the membrane. And what this protein does is it captures the antibiotic molecule before it can reach its target and then pump it out. And I say pump it out because this process is energy dependent. So the cell has to use some energy to pump out these proteins. And one of the features of these proteins is that they have really broad substrate specificity, which means that one protein can pump out any molecule that you can think of, any molecule of antibacterial nature. So just overexpression of one protein in these resistant isolates will give you multi-drug resistant phenotype. And this is exactly what happened in that patient that I was talking about in the previous slide, that one protein was overexpressed by cells, and all of a sudden it was just resistant to every single drug that we had at our disposal. We have the protein structures now, crystal structures of these proteins, and in gram-negative bacteria, they form a channel like this, where the drug molecule can be captured from here and then pumped out. So it's a very smart way of evading the action of antibiotics, because all you do is, or the cell does, is captures the molecule and just hurls it outside the bacterial cell. What we think is that efflux pumps, because they are responsible for resistance to so many different classes of drugs, if there was a way to inhibit the activity of these pumps, we can probably revive a lot of drugs that are currently useless because of the activity of these. So that's one of the focus that we have in my lab. So what we're trying to do is we are trying to characterize functionally these pumps, see what kind of drugs they pump out. We are looking at the mechanism of regulation, how the expression of these pumps is regulated, because we need to know if within a patient, if the expression of pump can go up upon exposure to certain antibiotics. We need to understand that mechanism better in order to be prepared for what to expect. And then we are also creating some genetic tools which will allow us to study these pumps better. So what we did was to understand the role of efflux pumps in Acinetobacter, we looked at the genome of Acinetobacter and we found 6 different genes that encode for, or that may encode for efflux pumps. So we cloned all of these genes, and then we tried to see if they pump out clinically relevant drugs. And I won't go into the detail of this, but what I'll tell you about this figure is that we have created a system in our lab where we can take a gene, and all these genes, especially the drug resistance genes, are very tightly regulated in bacterial cells. It's almost like a light switch. And they don't have the switch on all the time. They only turn it on when they need to. So what we have done is we have created an artificial system where we provide that switch for the expression of these genes. So we put the gene under a regulatory control, and then we put the gene in a surrogate strain, a strain which is susceptible to antibiotics, and then we turn the gene on. When we turn the gene on, then we look for changes in the susceptibility of that particular surrogate strain, and that gives an idea what kind of drugs are pumped out by these efflux pumps. So just to give you a quick snapshot, we characterize this one pump we're calling ADE-FGH from Acinetobacter, and you see that this here is when the gene is turned off, this is when the gene is turned off, and these are the 3 drugs, chloramphenicol, trimethoprim, clindamycin, that we actually found that as soon as you turn the gene on, the resistance goes up. So higher number is indicating higher resistance, and we see that the resistance can go up to 64-fold. So this tells us that this particular pump pumps out these 3 drugs. The other advantage of this system is that we can use this for screening of inhibitors. So what we did was we turned the gene on, and then we added this inhibitor to our bacterial cells, and we see that the resistance levels, even when the gene was turned on, went back to when the gene was turned off. So this tool, this genetic tool, can be very useful in screening for inhibitors. So here what we are suggesting is that maybe instead of looking for new antibiotics, we should look for molecules that inhibit these pumps and just try to recycle the antibiotics that we already have. So we're not suggesting that this as an alternative to the search for antibiotics, but we're just suggesting this as another means for looking for novel antibacterial search. And then we have actually looked at clinical isolates of Acinetobacter from Canadian hospitals. This work was done by my PhD student Dinesh, who likes Thai food a lot. So we have some collaborators in University of Manitoba and National Microbiology Lab, and what they do is they collect different isolates, different organisms from ICUs of different hospitals. So they sent us a bunch of isolates of Acinetobacter from different hospitals. And then we screen these for resistance, and this is just a heat map, so it's actually not telling you whether it's susceptible or intermediate or resistant. What it's telling you where the MIC values are high. So you can see that pretty high MIC values for, and all of these isolates are showing resistance to at least one antibiotic. So then what we wanted to see was if efflux pumps were playing a role in resistance of these antibiotics, and we used a technique called real-time PCR, where you can measure the expression of genes, and expression of genes will, by measuring the amount of RNA, and that amount of RNA tells you how much of protein is present. So we looked for expression of this one pump, ADEB, we looked at ADEJ, and then we looked at ADEF. And to our surprise, one thing that we found among Canadian isolates was that ADEF pump was expressed most dominantly in these isolates, which is very different from what we see in European isolates. In European isolates, we see ADEB pump, which is the predominant pump. So this is one slight difference that we see in our isolates, and we really don't know why that is happening. And then we wanted to see if we could correlate this expression of pump with antibiotic resistance. And what we see is, especially for tigecycline, we see a pretty tight correlation with ADEB pump. So by just looking at the expression of ADEB, you can actually predict the resistance to tigecycline, which is, if you remember, is the drug of choice if you have an MDR isolate. So tigecycline is never used as a primary drug, but if you find an MDR isolate, that's when it's used. And we see a pretty tight correlation. So these two isolates, which have high resistance to tigecycline, are also overproducing this pump. The other thing that we are looking at, and this work was done by Andre, who's my master's student, actually ready to defend now, was how environmental conditions affect the expression of these pumps and thus the MDR phenotype. So let's say that, let's think about Acinetobacter. And Acinetobacter baumannii it was earlier thought that it was a common soil organism, but it's not. Acinetobacter baumannii hasn't been isolated from soil ever. So it seems like this organism actually evolved to survive in hospital settings. It's very, very well adapted to survive on dry conditions, low temperatures, and then that's from where it can jump into humans and cause infections. So one thing that we wanted to look at was if changes in the environmental signalings was somehow contributing to the resistance of this organism. So let's say that organism is sitting on this tabletop in a hospital, and well, it's kind of cool here, but let's say 25 degrees, and then it jumps into human. The first change that the organism will experience is change in temperature, 25 to 37. So what we wanted to see was, does this change in temperature contribute to the expression of, in one way or other, of these pumps and thus resistance? And we found some really interesting results. So let's look at this bar right here in the middle and the white bar. This is data from 30 degrees centigrade, and this is data from 37 degrees centigrade. So you see that these three genes right here, these are the efflux pump genes, and as soon as you bring the cell from 30 degree to 37 degree, pump expression goes up dramatically, which would indicate that the resistance would go up dramatically. Now, if you remember, the resistance by efflux pumps doesn't come for free for the bacterial cell. It has to use some energy. So this is a very smart mode of survival where if it's at a lower temperature, it doesn't have to express these proteins, doesn't have to use its energy to survive, but as soon as it gets into our body, change of temperature starts expressing these proteins. Whether or not this actually word of caution here is that this is in vitro analysis. So whether or not this actually translates in a human body is yet to be seen. We looked at several other factors, but another factor that I'd like to talk about is iron. So we all have iron, but our iron is just for us. It's always chelated, it's always bound, so bacterial cells really don't have access to free iron in our body. So when a bacterial cell moves from outside external environment to our body, another change that it experiences is lack of iron. And we wanted to see if lack of iron was somehow contributing to expression of these pumps as well. So if we look at this yellow bar here and the green bar here, this is excess iron conditions, and this is iron limiting conditions. So again, very dramatic change in the expression of these pumps. As soon as you take iron away from these organisms, they start expressing efflux pumps. So this way, the organism is very smart because it senses changes in the environment. It somehow figures out that, oh, I'm not on the table anymore. I'm actually inside somebody. Let me just start pumping up drugs because I'm sure they're going to start showering drug on me. So I have to start expressing pumps. So if I was a bacterial cell, I would think that I was pretty smart. And this is what we are dealing with. We are dealing with cells. We are dealing with organisms that are very, very smart. They are rapidly evolving. And we have to, the challenge is to stay ahead of them all the time. And that's one of the big challenges when we are dealing with antibiotic resistance. The other organism that I'll briefly talk about is Pseudomonas aeruginosa that we work with. And Pseudomonas, we know, is notorious for causing lung infections in cystic fibrosis patients. So this is a postmortem cystic fibrosis lung, and all this greenish tinge that you see is because of growth of Pseudomonas. It causes pretty nasty infections in burn patients, and then it produces a lot of different enzymes that can, like keratinase, which can actually melt cornea within 24 hours. So using Pseudomonas aeruginosa, what we are trying to understand is how these proteins come together and work as efflux pumps. So we are trying to understand the interaction between different components of this R&D pump, and this work is being done by Sarah Warren, who's another master student in the lab. And the idea here is, if we understand how these components interact with each other, we could probably find a way to inhibit the interaction, make this pump inactive, and then use, again, the same drugs that are out there for us, but not usable because of resistance. So this is what I'm going to talk about the work that's going on in our lab. If anybody is interested about knowing more about that, I'll be happy to talk to you after this. And finally, I would just like to come back to the cause of resistance. What is contributing to the resistance in these organisms? So the first thing is misuse. Misuse of antibiotics is a huge problem, not only in developing countries like Ivory Coast here, where you can actually buy your antibiotics off the street, but also in developed world like ours. There was a study from earlier this month where this was an American study where they said that of all the flu cases where antibiotics are prescribed, only 20% are of bacterial origin. So we have this huge problem of over-prescription in developed world versus no prescription in developing countries, or self-medication. So that's a huge problem. And when you look at the usage of antibiotics, there's a very clear trend of increased usage. So we're looking at increased usage here, and we're looking at resistance here. You see that more is the usage, more is the resistance. And we are actually lying somewhere in the middle here. So increased usage of antibiotics does lead to increased resistance. The other issue that a lot of times we don't think about is non-medicinal use of antibiotics. 80% of antibiotics produced in US or consumed in US are used for non-medicinal purposes in agriculture practices, in farms, and this data is actually from Europe, which is indicating how much of different classes of drugs that are used for non-medicinal purposes. These are not used in hospitals. They are actually used on farms and in treating crop fields. And so what's happening in Canada? Some of you might have seen this Marketplace documentary. This was televised last year, where they actually went around looking at poultry meat, different brands of poultry meat from different stores, and they looked at organic kind, non-organic kind, and they found that every single brand that they tested had detectable levels of antibiotics in them. So every single brand of poultry that is being sold today here to us contains some antibiotic. So what they did was they actually did something really interesting. They followed up, and they went to a farm in Quebec, which was selling organic poultry. And it was true, they were not using any antibiotics in the feed. So where were these antibiotics coming from? Turns out that they were buying their eggs from somewhere else, and those eggs already had antibiotics in them. So even when you, so when it comes to question of what is organic, what is non-organic, it's a difficult question to answer, because you may not be using any antibiotics on your farms, but it's everywhere. It's just everywhere now. So it's a really widespread problem. And then the other issue is that resistance does spread. So the question is, well, okay, we saw that antibiotics are not used are used a lot in Europe for non-medicinal purpose, but we are across the pond, so we should be okay. But this is actually quite a fascinating film. This is showing you air traffic over a period of 24 hours around the world. So each dot is representing an airplane. And you can imagine how fast antibiotic resistance can spread. So here is the day line here. So as you see that it's morning time in Europe, all of a sudden traffic really increases. So right now the traffic is all going to Europe, and then as the day progresses, it starts coming back. It takes less than 24 hours for any gene to spread around the world. We know that because we have seen this with SARS. And spread of resistance is exactly the same mechanism. So we had, a couple of years ago, we found a new beta-lactamase in India, which was called NDM1, and now it's all over the world. It's being isolated from North America, from Africa, from Europe. And this is what is happening. And there's no way to control this. You cannot control the spread. I think it really have to start working at the grassroots level. The other issue, which is actually quite close to my heart, is, and some of my colleagues actually use antibacterial products in their offices. I always give them a hard time. But last time I noticed she still had it, so I'm not. You go to a grocery store now to buy household cleaning products, and you'll see that everything has some sort of antimicrobial in there. It's such a big fad right now because there's no evidence that it does any good in our houses, in our homes. But it's a problem. Try finding a non-antibacterial hand soap. And last time I was at Mark's work warehouse, they had everything, every single piece of clothing that you can think of, coated with antimicrobials, and generally triclosan, so t-shirts, socks, sweaters, jackets, underwears, anything you can think of was coated with antimicrobials, antimicrobial soap, antimicrobial toothpaste. So the question is, what advantage does it offer us? So let me show you some data. This is just one study. And I'm talking of households. I'm not talking about using antimicrobials in hospital settings. That's a completely different story. But in households, so this study was done in Boston area, and you see that, first of all, does it impart any kind of protection from infection? And this is a group that used antimicrobial products in their homes for a year, and this is a group that did not. And then when you look at the kind of health problems that they encountered, they were all the same, very, very similar number. So there's absolutely no protection when it comes to preventing disease. Well, then what is the problem? The problem is, especially if you look at these gram-negative organisms, and this is a different study, this is not the same study, by the way, you see that resistance rates in households that use such products is way higher than the households that do not use these. So what we are doing is we are actually creating selective environment in our homes using these products, which is promoting antibiotic resistance. It's very, very easy. We work a lot with triclosan, and it's very, very easy within 48 hours to expose a cell to triclosan and get resistance to fluoroquinolones, because the mechanisms of resistance are the same, because they all use efflux pumps for resistance. So triclosan selects for efflux pump over-producing strains, and that strain is now cross-resistant to antibiotics. So the question is, just to end, we have to be very, very careful about the fact that we do not go back to this. We simply cannot afford to go back to this. And one of the things that we need to do is a day like this, where we share what we know, and we share this outside this room with our kids, with our family, just to make sure that the problem is there. It's a great problem, and consequences are going to be very, very serious. So there are associations like this. This is Antibiotic Action based in UK. What they are doing is they are educating school children. They are actually educating our government representatives, which is really, really important because they are the ones who are making policies for us. Talking about the problem of antibiotic resistance and the risk associated with that. So with that, I'll end. These are the individuals who have worked in my lab, past and present, some postdocs, graduate students, and a lot of undergraduates. And these are the guys who pay our bills. And finally, I'll just plug this in. I do have a PhD position available, so if you know me. Thank you very much for your attention. And I'll be happy to take any questions if you have any.
Sorry, Lacey wants me to ask why you're so awesome, but my question was, what about the gram-positive bacteria? Do they have influx?
They do. Gram positives, gram negatives have efflux pumps, and actually efflux pumps are conserved throughout different species, so even we have efflux pumps. So that's why finding an inhibitor for efflux pump is not that easy, because you really need to find something that does not inhibit our efflux pumps, but inhibits bacterial efflux pump. But gram positive cells have efflux pumps as well.
Do you anticipate that you will see bacterial change of efflux pumps as you go through this process of turning them on and off, over time?
They may, and that's something that we need to look at. One of the issues with, with efflux pumps is that you just need one compound to maintain the selective pressure, because it's basically pumping out everything. So as long as, say, for example, if you have triclosan in the environment, which is a common product in household cleaning products, they can still be resistant to different antibiotics. So they may or may not, but we don't know yet.
You mentioned the use of inhibitors as a possible adjunct to antibiotic treatment. Since you just mentioned that even we have efflux pumps, is that going to be a difficulty in the research to differentiate between inhibitors that wouldn't have an adverse effect on us?
It is. It definitely is. The inhibitor that we tested in our lab, when it was discovered, it was very exciting. But turns out that every animal model that tested actually ended up dying because of that inhibitor. So that's a challenge. That's definitely one of the challenges when it comes to finding inhibitors. Toxicity is going to be a big issue. So it just makes it even more difficult.
So I think you can see how lucky we are at UIT to have Dr. Kumar with us. He knows his science and he can tell us what it means to us in our everyday life. So thank you very much.